And Steph, you say the sound is good? You can give us a chat. You can hear us okay?
Good afternoon, everyone, and welcome to the NCDS DataBytes Lunchtime Webinar Series. Today's speaker is Kimberly Rabowski, and she is a Senior Translational Research Scientist at RENCI, a research institute at UNC-Chapel Hill. She's also an adjunct professor in the UNC-Chapel Hill Department of Genetics. At RENCI, Kimberly supports best practices for cyber infrastructure and new business development, especially in the domains of biomedical and genomic initiatives. Kimberly's publication topics include analysis of whole genome and targeted sequencing data via phasing, haplotyping, and variant interpretation. She received her PhD in bioinformatics from Boston University on a research fellowship from George Church's lab in the Department of Genetics at Harvard Medical School. Today, Kimberly is going to talk about clinical decision support systems with integrated genetic testing. And the talk will last approximately 40 minutes or so, and we'll have time for Q&A at the end. And the way that we handle Q&A is we ask you to type your questions into the chat box. We'll hold them to the end, and then I'll read them back to Kimberly, and she will answer them. So without further ado, thanks very much for showing up, and welcome, Kimberly.
All right, hello, and thank you for that introduction. I appreciate it. So today I'm going to talk more specifically about a technology developed at RENCI with the collaboration of a lot of different people that I'll show it in acknowledgement slide at the end. And this technology is called the Clinical Decision Support System, or CDSS. And today I'm going to talk to you, I'm going to start out by setting up the motivation for why we developed this technology, and then I'll talk to you about some competitive technologies out there, give you a deeper dive into the CDSS system itself, and finish up with future directions. So the first question is, what are the use cases for genomic data? Why are we creating clinical decision support systems to include genomic data? And there's a lot of very compelling use cases, both in research and in clinical. On the research side, we're very interested in understanding simple or Mendelian diseases and complex disease risks, such as diabetes and cardiopathies. We're also interested very much in biomarker development for understanding how genetics might affect drug metabolism and finding new drug targets. And so these are all really compelling reasons for incorporating genomic data into your clinical workflows for research purposes and for clinical purposes. If you have a patient walking into your office, you may like to use genetic testing data to rule in and rule out certain diagnoses. Also, more and more in oncology, genomics are driving treatment plans, and we're even seeing the emergence of more risk factor analysis using genetic testing. So before I go any further, just very, very briefly run over some fundamental nomenclature here. When you hear me talk about a base, I'm talking about a DNA base or a nucleotide. This is the A, G, C, and T. When I'm talking about a gene, this is a distinct sequence of DNA bases. These are the things that essentially drive human traits. And when I talk about an allele, this is one of two alternate forms of a gene. And when I'm talking about genetic assays, generally you'll hear the term of whole genome analysis, which is all the bases in your genome. When you talk about exomes, we're talking about just the coding bases in the genome, or just the genes, which make up a very small portion of the whole genome. And when we talk about targeted panels, we're talking about just some of the genes. You'll also hear me talking about variants, and when you hear me say variant, I'm referring to a genetic variant, which is basically an allele that differs from the so-called reference genome. So that's what a variant is. And when we talk about variant significance, generally we're talking about a variant in your genome that is either pathogenic or benign, and that basically tells us if it's significant, it's because it's linked to a certain disease or pathogenic, and if it's not significant, it's benign, it's not linked to a disease, and it's of less interest to a clinician. You'll also see variant types such as SNP or SNP, which stands for single nucleotide polymorphism. That's basically an allele that only is one base in length. You'll hear us talk about frame shifts, which is a very deleterious type of a variant that shifts the entire gene and changes the whole meaning of the gene. And you'll hear us talk about indels, which is short for insertion deletion. And we'll also talk about variant types in the context of amino acids that make up the proteins that are encoded from the genes. And when we talk about those variant types, we have synonymous, missense, and nonsense. And synonymous is a difference that doesn't really affect the amino acid. Missense might change the amino acid, but nonsense change the whole gene. So hopefully some of these terms, many of these terms will already be familiar, but if there are any that are not familiar, we've covered them briefly here. Okay, so the broad problem statement, the real motivation for behind implementing this technology, the CDSS, is to solve this problem that end-to-end clinical workflows are very complex, and they have a lot of custom use cases, and depending upon how you're implementing your workflows, whether you're an academic hospital or a CRO or a reference lab, or you're running clinical trials or clinical studies, you're going to have organization-specific requirements for those clinical workflows. So finding software to implement all this and to integrate your genomic variant interpretation with the electronic healthcare records is very challenging, because one of the reasons for that is that genomic data are very large and the analysis are very demanding on computational resources. And so typically you would need to integrate your pre-existing clinical workflows or newly designed workflows with a high-performance compute system. And on top of that, the whole area of genomic variant prioritization and reporting of incidental findings is not a solved problem. And so, you know, you may hear on the radio commercials that talk about finding your ancestry through genomic testing, or finding out different traits such as caffeine processing. And it sounds like it's a solved problem, but it's actually not a solved problem, which really kind of exacerbates the situation. And this publication that came out last year from some of the significant thought leaders in the field, including Heidi Rehm and our own Jonathan Berg, who is in fact one of the thought leaders behind the CDSS, published this paper last year that basically compared a number of laboratories, 9 labs, and how they classified genetics, the genetic variants as pathogenic versus benign. And you'll see that, you know, in the top figure, the top histogram, what that's showing us is that about 80% of the variants, the genetic variants coming out of these 9 different labs, were all determined to be the same, either, you know, disease causing or not. But about 5% had disagreement of the variants coming out of these labs. Some of the labs said that they were associated with diseases. Some of them said they were unknown. So having that kind of disagreement can have an effect long-term on treatment and other factors. And then the bottom graph, histogram, shows that the problem is even worse when you compare to ACMG recommendations, ACMG being the American College of Medical Genetics, who puts forth a guideline on a number of variants and how they should be classified, and how these labs are interpreting those, even those variants. There's still a lot of significant disagreement. And the reason why we have this problem and why there's so many issues and disagreement in interpreting these genomes is because, well, for one thing, we don't really fully understand what the human genome does. The human genome and even other organismal genomes do. And we're in a state right now where the knowledge base is really changing very rapidly. And the reason for that, of course, is that the genome is really big, and that's why this is also a big data problem, and one of the reasons why I'm talking to you about it today. The genome is over 1 billion bases. It's got over 20,000 genes, and there's a variant at about every 1,000 bases from the human genome, and many of those are novel. And then when you look at these variants, how do you determine if it's disease-causing or not? Well, if it is a variant that is well documented by the FDA and other regulatory bodies, then it falls in the bin on the left there. Yes, it's definitely pathogenic. We know it causes disease. But if it's not documented, then there's a number of different algorithms that can be used to determine whether or not it's pathogenic. And you can look at the right in this graph. We have moving on the x-axis from left to right, less and less seemingly deleterious types of variants, with intronic variants being those variants that fall within, fall outside of the coding regions, fall outside of the genes, are thought to be really not deleterious, although they still may be. And on the left, the nonsense or splice site or frame shift variants are thought to be very, very disruptive in the coding portions of the genome and can really affect your proteins, and so are thought to be more pathogenic. However, we still see these kinds of variants in high frequencies in the population. So that's why on the y-axis we also consider allele frequency when we're binning these variants between pathogenic and benign. And so if you see something that is a nonsense variant, it should totally disrupt the gene. But if you're seeing it at a frequency of, say, 99% in the population, then it's very unlikely to be pathogenic. So these are the kinds of things that drive how people today are trying to interpret the genome and incorporating these features into the clinical workflows. So now I'd like to just pause a moment and tell you about what kinds of clinical genomic providers are out there. There's a lot of software, a lot of databases, a lot of different things, technologies to choose from. And broadly, you can bin them up into those that provide genomic variant annotation. If you need variant annotation, nothing else, then maybe you just look to those. If you need to incorporate those annotations into an analysis pipeline, there are providers that give frameworks for doing that in the cloud. There are providers that give the entire software system for clinical workflow management all the way through to reporting. And those would use, again, analysis pipelines and annotation, so each one of these steps building upon the previous one. There are providers that give the full service. You just send them samples, and they give you back a report. And finally, there's the direct-to-consumer providers that you don't even need to go to a doctor's office. You can just order the kit online. And so, again, here are those different categories mapped to individual providers in each column. Starting on the left, we have N of 1 and Watson, which are both, especially Watson's probably more commonly known for Watson Genomics and Watson Oncology, providing just the annotation. And these providers tend to team up with other providers to beef out and add on to the annotation the analysis. And so you can look to providers like DNA Nexus, which is the predecessor to DNA Nexus actually is BaseSpace, which is hosted by Illumina, and both of these and some others provide that kind of cloud-based analysis pipeline. Clinical workflows takes you from the sample all the way through all of your patient management and integrates your genetics and everything else. And providers, a big player in the field there is Purion DX. This is also where CDS fits in, the technology that RENCI has. And then there's a number of full-service platforms and a number of direct-to-consumer platforms where the direct-to-consumer is probably the most famous one might be 23andMe and Ancestry, and there's a couple of others. And this is not meant to be a comprehensive list, but just to give an idea of what the different things are out there. So we're going to talk about CDSS. And so CDSS is the open clinical decision support system, and it manages everything from patient intake and scheduling. It can manage consenting and all the way through to clinical reporting. And we also include genetic testing. We have end-to-end workflow auditing to support different regulatory needs such as CLIA validation and Part 11 compliance, and at the end we also include automated clinical reporting. Some of the users of this system you can see here on the right, and the applications include whole genomes. We can do whole genomes. We haven't processed whole genomes, but technically we could. But most of these users have used it for processing exomes and targeted panels. And the way the targeted panels work is that a gene list is provided to the system operators, and an exome is processed through the lab, and only those genes that are of interest then are exposed to the clinician. We've also, ClinGen has used this system for variant curation in some incarnations, and germline risk alleles are integrated. The system is integrated with UNC-CH for finding the risk alleles in oncology pipelines. So the high-level functionality, again, this is fully configurable because it's freely available, it's open. It provides monitoring for patient intake, sample collection, and lab assays, parallelized analysis report sign-off, soup to nuts, right down to the automated clinical reporting. So this is a screenshot. You're not intended to be able to read it, so don't strain your eyes. But this just gives you a feeling for one possible view that could be created using this framework. This view gives us everything for the participant, all the details from demographics through all the workflows, the lab workflows, the clinical visits, what's scheduled, what's passed, inventory, biosamples, and all the way through to the HPC integration, showing the quality assurance results from the sequence analysis pipeline, and as well as timestamps and clinical notes from any clinicians that have added those things. So you have everything in one place. I just want to point out that for this screen, this is looking at the Coriell provided publicly available sample NA12878. So obviously there's no PHI being presented here in this talk. And in fact, actually in this system, it's designed to operate external to PHI. So by the time you get here, all that information is stored in a different HIPAA compliant situation. Okay. So for this slide, we're looking at the high-level architecture and all the different components that make up this system in terms of the implementation. And what I'd like to do is talk to you in terms of the functionality that is implemented on top of each one of these components. And so starting with the molecular analysis view, which is supported by our Canvas system that provides genomic interpretation using variant annotations and the observed variants. And so the molecular analyst view may look like this, which basically, again, don't strain your eyes, but this is just a table of variants with a lot of annotations attached to them. And so zooming in on one row, you can see that some of the information provided using this framework, you can present the variant effect. The class, in this case, this variant is being specified as a variant of unknown saying why they think that this thing is actually pathogenic or it's not, maybe, again, is unknown significance. And there's a lot of other information on this screen that can be revealed to give more context and allow for more diving into this particular variant to really try to understand it, linking out to things like OMIM, which is an external resource for looking at genes and diseases, or linking out to the UCSC genome browser so that you can see this particular variant in the context of the whole genome and other annotation tracks. So that's the molecular analysis view, excuse me. The other important component I'd like to briefly go over with you is genetic variant annotation release management, because doing these annotations requires pulling in data from a number of different sources. A lot of external data sources don't do very rigorous release management, but you really do need to version these different annotation databases, whether it be by using the external versions or creating your own versioning. And so as you can see here, there's a number of different annotation external sources that come into play. There are gene-specific ones like Ensemble, RefSeq, and ENCODE, and there's variant-specific external annotations like dbSNP and polyphen, which is actually algorithmic annotation, but nevertheless a third-party provider for that kind of information. So there's a number of different sources, and so how do we do strict versioning on this and make sure that we stay fresh with the most current data for our annotations? We've developed a framework we like to call AnnoBot, with our little Robbie robot there on the right to help you remember it. But basically, the AnnoBot goes through different modules, one per each of these reference databases, such as dbSNP, for example, for Module A, and it looks at the external data source to see if there's something new, and if there's nothing new there, then it goes to the next module. And if it finds something new in the next module, in this case, for example, RefSeq, then it will go out and ETL the data and update its database with the versioning information and timestamp and everything else. And so this is how we make sure that we have the most current annotations, and also the reports get tagged with the annotations so that all of the interpretation is reproducible and repeatable. And so that covers that area of the architectural diagram. Now, I have this area here that shows the integration with the next generation sequencers, the high throughput genetic sequencers that write to an IRODS data policy manager, and I also have on the diagram here the MapSeq component, which is the scientific workflow and the cluster manager that allows you to take advantage of local and external clusters. And this whole portion of the diagram shows how we can integrate HPC into these clinical workflows. There are white papers that I'll give you links to at the end of this talk that discuss both of these in greater detail, so I'm not going to go over that here. So the next thing I want to tell you about is the GMW, which does the full orchestration and presentation of all these different views, and it integrates with the lab instruments and the electronic health records and the legacy systems, including user management, such as Active Directory, and it also caches a lot of the data that it gathers from all these different areas into a non-PHI data mart for rapid retrieval and presentation. And so this screen is just one of the views under the administration, showing how you can achieve easy user management, again, all from one console, providing roles to the different users. This screen is showing how the integration of your wet lab assay auditing. This is the wet lab dashboard, and there's a number of different views off of here. I just wanted to give kind of a taste of what's possible. And from here, you can implement scripts for the robotics in the wet lab and look at your pooling in a lot of different nice interfaces from there. And then this view gives you a view into the different analysis workflows and shows how you can actually see that your analysis is moving along nicely and can get an idea about when it will be done. So I've talked to you about this, the entire architecture and the next steps for the CDSS, where we are in the process. We've already developed a piece called, we like to call Karnak, for further understanding the variant within the context of the genome. And so this is a nice little sneak preview into Karnak. It's not actually in service at the moment with any of the CDSS use cases. In fact, we have processed thousands of samples through CDSS and are currently processing about 1,000 medical genetic patients' samples through CDSS, but Karnak is not integrated into that pipeline at the moment. So this is just giving you a preview, and looking at the figure on the right here, on the top is shows how you'll be able to have view customization. This is already implemented. As I said, it's just not, it's still in alpha testing. The box circled with B is showing you your gene summary information, and on the bottom of this view, in the box, I'm going to jump down to E, shows you the variant of interest, where the variant itself is colored by the severity and the effect, whether this is pathogenic or considered to be benign, and what it's actually doing, if it's a frame shift or how, in a missense or nonsense type mutation. So you get all that information from color and shape pretty quickly, and you get it in the context of all the other gene transcripts that might overlie that particular location. And on the top box, C, you also see these colored towers, which give a visual representation of the allele frequency, how often that particular variant is seen in the population. So that, again, if you look on the bottom in box E and you see your variant has a very severe prediction and potentially a very harsh effect, but then you also look at the top boxes, C, which shows you the allele frequency, and it's very frequent in the populations, it may make you choose to decide that, well, you don't really know if this is pathogenic or not. Okay, so these workflows are complex. They require a lot of customization. The thing that makes CDSS unique in the market is that it's open. The other big player probably in the field is PeriodDX, which is not an open system, but still can be configured using consulting services from PeriodDX. But if something like the open systems for CDSS sound interesting, or if there's some portion of it that might be usable in your workflows or your colleagues' workflows, we would encourage you to reach out to us. We can help with providing customized demos of the software. We can help with requirements analysis and systems design specific to your legacy systems. We can help implementing if there's some feature that is really interesting to you and we don't have it yet. We can look into implementing that for you, or help with testing and deployment. And we're also available to provide support for remote configuration, integration and installation, and also to provide training and upgrade type sustaining services. And that's all. Okay, I'm bouncing around a little bit on the slides here. I apologize for that. So that's all I have for. Where are we? Oh. That's all I have to talk about for you today. And I just wanted to finish up with the acknowledgments. We have a number of white papers here. If you're interested in learning more about the HPC integrations with MapSeq or with iRods, I would encourage you to take a snapshot of this screen, and or you can reach out to me at krabowski@renci.org, and I can forward those links to you. But as you can see, this is a project that's been, had a number of releases now, and it's been in use in UNC hospitals and for clinical studies, and has really had a lot of people involved in it. And I'm very grateful to all these participants for providing their feedback and improvements over the years of the many incarnations of CDSS. Thank you. Okay.
Kimberly.
I'm going to
Okay.
throw this.
We're pulling up some questions here for Kimberly. Kimberly, thank you for that presentation on a really great collaborative project. And if you can bear with us a second while we pull up questions that have come in the chat box.
Yeah.
Can I just do them here?
Yeah. We'll just leave it like that.
Okay. Is that it?
Mm-hmm.
Where? I don't see the question.
That was just from staff.
Okay.
Oh, here's one.
Okay. I'm sorry. We're having a little bit of technical challenges today, but okay. So first question. What was the motivation behind creating the CDSS? Given that there are other genetic testing platforms, what makes CDSS unique? This is a multi-part question.
That's an excellent question.
And there's more to it, but I'll let you get to that first part first.
I mean, the biggest difference, so understand that CDSS has been around for a number of years. It predates a lot of the systems out there on the table that I presented and a lot more that I didn't mention. And so the motivation initially was there was just nothing there. But over the years, it made sense not to replace it, you know, as I said before, primarily because it isn't a completely open system. The source code is freely available and customizable. And for an academic hospital such as UNC hospitals, that makes a lot of sense when you have resources on staff that can quickly make the kinds of customizations that you need. It's not the right solution for everyone. You know, you've heard me mention probably the closest thing to CDSS is PerionDX, and PerionDX is fantastic because you can actually get a clinician in your workflow. If you're a reference lab, for example, and you don't have a medical person to sign off on your clinical reports, then you might want to go with something more like Perion. But the other thing that CDSS offers you is support for a lot of additional things, like I said, writing the scripts for and exercising the scripts for the lab instruments, and a lot of those things that are kind of upstream of where you come into Perion. So this is really, you know, for the lab that's looking for something that, you know, they can do a lot more customizations. You may have a lot of legacy systems that are, you may have some unique challenges, and CDSS might be for you.
Okay, and then related to that, do you see software players in the field adapting their services in the future? For example, will Watson continue to sit at one end of the service providing spectrum, or do you see it getting into direct-to-consumer reporting later on?
So the system that I presented is really just software, and when we talk about direct-to-consumer reporting, I suppose if you had a startup and you wanted to start a direct-to-consumer business, then you would find vendors for managing your samples in terms of, you know, packaging tubes and that sort of thing. And then you'd want to buy software that you would want to CLIA validate end-to-end, and that software could be CDSS. So that's how that fits in there. So we would not personally provide any direct-to-consumer reporting, but potentially someone could use this technology to do that. And if the next question, that may beg the question of, is UNC Hospitals going to provide direct-to-consumer reporting? I have no comment on that. I have no idea.
Okay, any other questions at this time? We have a little bit more time to take some. No? Okay, please remember that we post the DataBytes webinars online. It usually takes about a week to get them online, so check back on our website, which is datascienceconsortium.org for that. Once again, Kimberly, I want to thank you for your time today and for presenting this time. We appreciate your time, and we appreciate our attendees who have taken their lunch hour to listen to us. And everyone, please remember that DataBytes is a monthly lunchtime webinar series. We usually hold our webinars on Wednesday, and we'll be back to that regular Wednesday schedule next month, Wednesday, May 3rd at noon, and our speaker will be Angela Zoff, Data Visualization Coordinator at Duke University. So we hope you can join us then. Again, we thank you for joining us today. We'll let you know when the webinar is posted, and everyone have a wonderful rest of the day.